Chapter 16, Sensory, Motor, and Integrative Systems. We start this chapter by talking about sensation. Now for sensation, we're going to look at the actual definition of sensation, which is the conscious or subconscious awareness of changes in the external or internal environment. So this is the physical detection of stimuli, whether it be tickle, itch, hot, cold, pressure, increase in blood pressure, physiological changes within the body, chemical changes in the body, increase in pH or CO2 or decrease of O2. So these are things that are sensed. So again, conscious or subconscious awareness of these changes. Perception is going to be a little bit different. The definition is the conscious interpretation of sensations performed mainly by the cerebral cortex. Now perception can vary. And what I mean by that is that we perceive sensation differently from person to person. A good example is tickle. Some of you may be extremely ticklish. Just hearing the word tickle could elicit a response, whereas some of you might not be ticklish at all. Now, does this mean that you have less receptors to detect the tickle? And the answer is no. We all have roughly the same receptors for detecting these senses. The difference is going to be how we perceive that stimulus coming in. Let's elaborate on the sensation. Each type of sensation is called a sensory modality. Things that we can detect are touch, pain, vision, and hearing. Sensory modalities are grouped into either general senses, this includes touch and pain, or special senses like vision and hearing. Our general senses, again, somatic, tactile, thermal, proprioceptive, balance, visceral pressure, chemical stretch, nausea, hunger, and temperature. Our special senses include smell, taste, vision, hearing, and equilibrium. Now, different types of sensory receptors are involved in the different senses. For example, when we have a cold stimulus coming in our first order sensory neuron, we have these free nerve endings that are going to elicit a receptor potential. That's going to trigger the nerve impulse to fire down the axon length. It propagates to the central nervous system, at which point it will be translated. Now, just to bring back in AP1, where we talked about a unipolar sensory neuron. So remember, when we talked about our unipolar neurons, we said that typically they were sensory, and here's a good picture of that. Now, the free nerve endings we just talked about are usually used to detect pain, temperature, tickle, itch, and some light touch. We find these close to the surface of the skin. Their nerve endings, as we can see here, the reason why they're called free nerve endings is because they are not encapsulated in any type of fat or capsule. They're just out and about into the interstitium or tissue. And in the case of smell, they're just out into the air, embedded in this mucus patch in the uppermost portion of your nose, which would allow for an odorant or a stimulus to be detected, an odorant to be dissolved in that mucus and then fire the receptor potential. The second type of nerve endings are encapsulated nerve endings. These are usually encapsulated with fat. They're used to detect things like constant pressure, deep vibration, and deep touch. So a vibration stimulus comes in. Again, we're talking first order sensory neuron with that encapsulated nerve ending. A receptor potential is generated. It's triggering this nerve impulse down the length of the axon, which will propagate into the central nervous system. Lastly, sensory receptors for some special senses include gustatory receptor cells, so taste and taste buds, photoreceptors in the retina of the eye, and hair cells in the inner ear for hearing use a receptor cell to generate a receptor potential. So we start with the sensory receptor synapsing with the first order sensory neuron. An example would be a sugar molecule coming in, exciting this gustatory taste receptor, which will cause it to release neurotransmitter from that cell, which will trigger this nerve impulse or action potential down the length of the axon, again propagating into the central nervous system. So this go-between, this receptor, the the part or cell that is eliciting that action potential down the neuron is the key difference to the two that we talked about before. Now we can also group receptors based on location of the receptor and the origin of the stimuli that activate them. For example, we have exteroreceptors, extero meaning outside. We're also going to have interoreceptors, meaning inside. Now those exteroreceptors include receptors that are detecting hearing, vision, smell, taste, touch, pressure, vibration, and pain, all things that are detected outside the body. Now if we go inside, as we alluded to just a minute ago, we have our interoreceptors. Now these interoreceptors are monitoring the body's internal environment, things like chemical changes in the blood, pressure changes in the blood, etc. The third type are proprioceptors. These are providing information about body position, muscle length, and tension, and the position and movement of joints. So overall, balance and continuity between muscles, bones, tendons, and ligaments. Table 16.1 goes through the basis of classification of all of our different sensory receptors and a basic description. So you could go through these. We've talked about free nerve endings, encapsulated nerves, separate cells, which include those receptor cells that are synapsing with those first order neurons. We've talked about extero, entero, and proprioceptors. What we haven't really talked about was naming them based on stimuli detected. So mechanoreceptors detect stretch, typically in the skin and in blood vessels and internal organs. Thermoreceptors detect changes in temperatures. Nociceptors are for pain. Photoreceptors detect light. This is for vision. Chemoreceptors detect chemicals, things like in our mouth for taste or nose for smell. Osmoreceptors detect osmotic pressure of body fluids. Somatic sensations. Somatic sensations include tactile, thermal, pain, and proprioceptive. Tactile sensations include touch, pressure, vibration, tickle, and itch. Let's look at a picture and an example of some of our receptors. So we look to the sensory receptors in the skin. We start with a type 1 cutaneous mechanoreceptors. These are known as Merkel's discs. Their job is to sense continuous touch and pressure. The next one we can talk about is Meissner's corpuscles. These are these whirlwind-looking structures. Their job is to detect touch, so they sense the onset of touch and low-frequency vibrations. Notice that these guys are close to the surface of the skin, so all initiation of touch and pressure and light, the onset, the beginning of touch. Next are Ruffini's corpuscles, these guys right here. These are type 2 cutaneous mechanoreceptors, whereas Merkel's discs were type 1 cutaneous mechanoreceptors. These guys are sensing skin stretching and more deeper pressure. The hair root plexus, we talked about this with the integument system, senses movement on the skin surface that disturbs the hair. So again, blow on your forearm, all that hair moves. You can feel that because of this root plexus. Pacinian corpuscles, these big thumbprint looking structures covered in fat, they are lamellated corpuscles. They sense high frequency vibrations and constant pressure. The last ones to talk about are our free nerve endings. Look how they surround that papilla. These guys are here to detect pain, itch, tickle, cold, or warmth. Let's focus in on pain. We have two types of pain, fast and slow. Fast pain is also known as acute pain. This is sharp or pricking pain, as if you received an injection or a shot or a needle. This is perceived within 0.01 seconds. So we're talking about a really fast pain. Slow pain is that chronic burning, aching, or throbbing pain. This is perceived a second or more after the stimulus. So, for example, if you burn your finger on a hot stove, it can ache for hours or days. This is slow pain. Now, superficial somatic pain is pain that arises from those skin receptors. Again, we use that word superficial, talking about the epidermis. Deep somatic pain is pain that we feel in our skeletal muscles, joints, tendons, and fascia. And last, we have visceral pain. This is stimulation of pain sensors in the visceral organs. Now, this is a little weird because we don't have a good way of localizing visceral pain. So as a result, this visceral pain is deciphered or felt in different parts of the superficialmost layer of our skin. And we're going to look to a referred pain chart to kind of explain this. Now, again, for visceral pain, it usually presents in or just deep to the skin that overlies that stimulated organ. Again, we'll look at a referred pain map to really explain what this means. So a quick diagram, quick picture to kind of explain referred pain. Now, again, the best way to explain referred pain is think about a heart attack. When you have a heart attack, where do you feel it? Chest, under your arm, down the medial aspects of your arm and forearm and into your pinky. And look at the red. That referred pain is pushing all the way down. So again, it's not really where the organ is, but to the closest plexus, and we'll talk about plexuses later on. We did this in AP1, so it should be a little review. And then pushing through that referred pain map. The heart, again, look, in the jaw. This is where a lot of places women feel this, as well as in their shoulder blades at the base of their neck. The kidneys, look at their distribution. We're talking coxal, lumbar, all the way through the hips, the anterior lateral aspects of the thigh, lower back, etc. So this is a huge referred pain distribution map. The bladder, sacral region, lower gluteal region, and lower femoral region, hypogastric regions, things like that. Okay, so again, look at the liver and gallbladder, the second largest organ, liver in our body, and it's taking up our right shoulder, even though the liver is localized down through here, right? So an interesting way to see how those deep organs, how we perceive pain to those deep organs. You know, a good example, look at our ovaries, right? Right here in that lower back abdominal region. So one of the most dangerous cancers for females is ovarian cancer. And the reason why is a lot of people say it's asymptomatic. It's really not asymptomatic. It demonstrates symptoms. Those symptoms are sometimes abdominal pain and or lower back pain. So now think about how many of you guys have ever suffered from lower back pain? Probably a lot of you, right? I doubt the first thing you thought of was ovarian cancer. That's why this cancer really goes undetected until the later stages, which makes it a really dangerous cancer for females because of the way our body deciphers and interprets deep visceral pain. We can continue with somatic sensations, talking about proprioception and proprioceptors. Proprioceptors are the actual receptors located in muscles and tendons, whereas proprioception is recognizing position of the body and its associated parts. Now let's look at some of those proprioceptors. We talk about muscle spindles. These are a type of proprioceptors found in skeletal muscle. They monitor muscle length and are involved in stretch reflexes. So if you've ever been to the doctor and they have taken that reflex hammer and hit, say, your bicep or tricep, they're looking for those muscle spindles, the proprioceptors firing, and will give you this arm jerk to check your stretch reflexes. We have tendon organs. These are types of proprioceptors found at the junction of a tendon and a muscle. They protect muscles and tendons from damage due to overstretching. Again, if you've been to the doctor and they take that reflex hammer and they hit your patellar ligament in your knee and you get that knee jerk, this is checking for those tendon organs, those reflexes. Joint kinesthetic receptors, these exist in and around the joint capsule of synovial joints. They respond to pressure, acceleration, and deceleration during movement and help to protect against excess strain. Maybe while you've walked, you hit an uneven terrain and your knee buckles on you. This is a type of reflex because of what's going on within that joint. So we can see the muscle spindle. Look at the nerves coming in from the CNS, touching those motor units, and then response going out. Okay, so the sensory neurons going out, motor response coming in. In our picture, we also see our tendon organs. So look at that nerve going into the actual tendons. These are these tendon reflexes versus muscle reflexes. Table 16.2 goes through all the different receptor types, the receptor structure and location. what they sense, and their adaptation rate. So this is important. We talk about sensory adaptation. This is going to be the first time we talk about it in lab. We're going to talk about this in a little more depth, but different receptors adapt to their sensory stimuli at different rates. For example, Meissner's corpuscles, the onset of touch, they adapt pretty quickly. Whereas those type 2 cutaneous receptors, those deep ones, they adapt more slowly. And this is good because this is what tells us that we're still holding on to that pencil or still holding on to that steering wheel while we are driving. We can look at pain receptors. These guys are slow to adapt, right? Our muscle, spindles, tendon, organs, joint kinesthetics. Muscle, spindles, tendons, slow to adapt, and joints are rapid to adapt. This is why when we're running or walking, we need a rapid adaptation. We see this in our skin too, right? So we get a rapid adaptation to our clothes on our body or that wallet in your pocket. That's why you don't feel it after about 45 seconds. Same thing with smell. It takes about 45 seconds for those receptors to adapt to that sensory stimuli coming in. We're trying not to. Why do we have adaptation rate? We're trying not to overwork and overload the brain with unnecessary and consistent information. Somatic sensory pathways. Now, somatic sensory pathways carry information from somatic sensory receptors to the primary somatosensory area in the brain, specifically the cerebral cortex, and then to the cerebellum. The pathway of the cortex consists of thousands of sets of 3 neurons classified as, and we've introduced this before, first order neurons, second order neurons, and third order neurons. First order neurons carry impulses from the somatic receptor to the brainstem or spinal cord. Now, second order neurons are going to take those impulses from the brainstem and the spinal cord and move them to the major relay center of the brain. These 2 egg-shaped structures called the thalamus. Now, that thalamus is that major relay center from first to second. And again, now, third order neurons are going to carry from the thalamus to the primary somatosensory area of the cortex on the same side. Now, this is a key note, on the same side. At some point during this process, we get a crossing over from left to right and right to left. This crossing over is called decussation of nerve. So remember this word. Again, we talked about this in AP1, so it's just a reintroduction, revisit of these basic terminology. The next step is to look at our pathways. So somatic sensory impulses ascend. So this is important. Sensory tracts ascend, motor tracts descend. So again, look for these key words. Somatosensory impulses ascend to the cerebral cortex along 3 general pathways. The first is the posterior column medial lemniscus pathway. The second is the anterolateral or spinothalamic pathway. The third is the trigeminal thalamic pathway. And again, the nice thing about these pathways is they tell us where they start and finish, right? Like spinothalamic, the spinal cord to the thalamus, trigeminothalamic. So we see the thalamus and the trigeminal nerve, one of our cranial nerves right here. Now, the posterior column medial lemniscus pathway is going to give us impulses from the limbs, the trunk, the neck, and the posterior head. And we could look at a picture of this. So we see receptors for touch, vibration, pressure, and proprioception in the lower limbs and lower trunk moving, ascending through the gracilis fasciculus and the cuneate fasciculus in the posterior column. It moves up to our second order neuron. We see it synapsing. And then, here we go. We came in on the right side. We decussate to the left. We come up to the medial lemniscus, up to the thalamus, second order, from the thalamus to the primary somatosensory, third order. First order, second order, third order neuron. The anterolateral or spinothalamic pathway takes impulses for pain, temperature, itch, tickle, and senses from the posterior head. We can look at a picture to see where the information is coming in. On, let's say, the left side of the body, receptors for pain, cold, warmth, tickle, and itch. There's that sensory neuron. Comes into the spinal cord, decussates here to the anterior spinal thalamic tract. It ascends and moves up through that spinal thalamic tract. First order here, second order here to the thalamus, and then third order from the thalamus to the primary somatosensory area of the cerebral cortex. Now, the trigeminal thalamic pathway takes impulses from most somatic sensations, tactile, thermal, and pain, from the face, nasal cavity, oral cavity, and teeth. Again, think of that cranial nerve 5, that trigeminal nerve, and what it innervates. Somatic sensory impulses reach the cerebellum via the spinocerebellar tracts. So we can look at a picture to see the information coming in. So we come first order neuron here, information comes in, receptors, touch, pain, vibration, pain, cold, warm, itch, tickle, face, nasal cavity, oral cavity, and teeth. We decussate here in the pons, and then our second order neuron, you see, we also go to the medulla oblongata. Second order neuron moves up through to the thalamus, third order neuron to the primary somatosensory region. So again, first order neuron, second order neuron, third order neuron. Now, we've talked about this primary somatosensory area. Think back to AP1. Remember, it is located on both parietal lobes on that post-central gyri. Each region in this area receives sensory input from a different part of the body on the opposite side. So what's being sensed on the left is really being integrated on the right, and vice versa. What's sensed on the right is being integrated on the left. Again, that word, decussation, the crossing over of tracts and nerves to deal with opposite sides of the body and brain. Now we look back to our homunculus, which tells us each point of the body maps to a specific region in the primary motor area. We will use a 2-point discrimination test in lab to kind of measure how much of our cerebral cortex is dedicated to specific areas. For example, we see the lips get a massive amount of cerebral cortex, whereas the entire trunk of our body is considerably less. So what does that tell us? The distance between nerve endings around the lips are closer. We are able to detect much more, or have a higher sensitivity to touch there, whereas our back, we're not dedicating as much cerebral cortex because it is not as important of a receptor or site of reception as the mouth. Look at the tongue. Tongue and pharynx and intra-abdominal region gets this huge amount, whereas our whole leg gets, a fraction, a third of it in comparison. The posterior spinocerebellar tract and the anterior spinocerebellar tract carry proprioceptive impulses to the cerebellum. These pathways are really important or critical for posture, balance, and the coordination of skilled movements. Now, table 16.3 outlines the different tracts and works through their pathway function. So for you guys should be working through the tracts, where their nerve impulses, so where their nerve impulses go, and tracing first, second order, and third order neurons, and where they decussate, if they decussate, because remember, not all of these decussate. Again, a continuation with that table, all the tracks, where they go, what they do, how they decussate, etc. Now that we have finished somatic sensory pathways, we can continue on with our somatic motor pathways. The first part of the somatic motor pathway is looking at nerves that extend out of the brainstem and spinal cord and innervate and move to skeletal muscle. This is where they terminate. These are called lower motor neurons. Again, lower motor neurons innervate skeletal muscles of the face and head through our cranial nerves, and skeletal muscles of the limb and trunk through spinal nerves. Our somatic motor pathways provide the input into lower motor neurons, LMNs, and are divided into 4 distinct circuits. The first are local circuit neurons. These are located close to lower motor neurons in the brainstem and the spinal cord. The next are upper motor neurons. These neurons input to both lower circuit neurons and motor circuit neurons. Ultimately, they directly signal muscles to contract or relax. Basal nuclei neurons assist movement by providing input to those upper motor neurons, and ultimately cerebellar neurons assist movement via control of activity of those upper motor neurons. Remember, these are going to skeletal muscles telling them to contract or relax. So let's look at a flow chart for our somatic motor pathway. We can start with our local circuit neurons in the brainstem and spinal cord. These activate lower motor neurons, which is our final common pathway, which talks to the skeletal muscle. Now, we can go back to, say, the sensory motor and association cortex, our upper motor neurons from the cerebral cortex that talk to the motor centers in the brainstem, which in turn, we follow these upper motor neurons, talk to those local circuits, those lower motor neurons to contract or relax skeletal muscle. We also see the basal nuclei coordinating that information with the thalamus and those cerebellar neurons, again, going back to those upper motor neurons, so basal nuclei to the thalamus, basal nuclei to the motor centers in the brainstem, activating those lower motor neurons, or dealing with the upper motor neurons, which will then turn activate those lower motor neurons. Now, our upper motor neurons extend to the lower motor neurons through two types of pathways. And one is a direct motor pathway. This is where we're delivering the signals to the lower motor neurons directly from the cerebral cortex. The second is an indirect motor pathway. So this is where we're delivering signals to the lower motor neurons from motor centers in the basal nuclei, dealing with body movement and coordination from the cerebellum and cerebral cortex. Now we could trace the pathways. There are two direct motor pathways used for voluntary movement. The first is cortical-spinal pathway. It's the lateral cortical-spinal tract and the anterior cortical-spinal tract, and the cortical-bulbar pathway. Now we can look at a drawing and we can see that we start, now we're doing the opposite of the sense. Remember, these are descending, so we're starting at the primary motor portion of the cerebral cortex. We're descending down through our cerebral peduncles, through the pons. We decussate, cross at the medulla, pass through the lateral cortical spinal tract, through our lower motor neurons, out to our skeletal muscle in the distal parts of the limb. So again, this is the cortical spinal pathway. We can look at the anterior cortical spinal pathway, follow the same track, primary motor area, cerebral cortex, down. We move through the pons, through our pyramids, into our anterior cortical spinal tract. We're going to decussate right here and then move to our lower motor neurons, which we will process out into the trunk and proximal parts of the limbs, again talking about skeletal muscle. The next is our cortical-bulbar tract. We start at the primary motor area. Our cortical-bulbar tract moves down through the cerebral peduncles, at which point we're going to hit the facial nerve on the right and left side. We move to our lower motor neurons, which will propagate to our skeletal muscle for facial expression, or we'll travel down to our hypoglossal nerve, cranial nerve number 12, move to the skeletal muscles of the tongue. Now for our indirect motor pathways, these include our extrapyramidal pathways, which are our rubrospinal, tectospinal, vestibulospinal, lateral reticulospinal, and our medial reticulospinal tracts. So let's start on top. We'll start at our superior colliculi. We follow it down where it's going to decussate and process through our upper motor neurons. We're going to continue down to our tectospinal tract, into the spinal cord where lower motor neurons will travel to skeletal muscles. We follow the red nuclei. We decussate, we move down upper motor neuron to the rubrospinal tract, again down to lower motor neurons and out to skeletal muscle. Let's take our vestibular nuclei. We move down to our vestibulospinal tract, hence vestibular nuclei. We process down to the spinal cord and out to skeletal muscle. We can take our reticular formation. Here we go, reticulospinal, right? So we start here, we move down to our reticular spinal tract, down to the spinal cord, out our lower motor neurons. Now, similar to sensory, motor is no different. We use our homunculus to explain that each point of the body maps to a specific region of a primary motor area. And again, you see that for motor, vocalization takes up this huge space, our mouth, our pharynx, whereas our elbow, wrist, trunk, shoulders occupies a relatively small amount of motor response for cerebral cortex. Again, we culminate all these pathways with the table showing you the pathway, what they do, how they descend, if they decussate. So again, look at the lateral cortical spinal pathway as an example. 90% of them decussate, cross over to the opposite side, and then enter the contralateral side of the spinal cord, and where they terminate with lower motor neurons. The anterior cortical spinal, the 10% that do not decussate enter the spinal cord and form this tract. So again, where they decussate, if they decussate, what they do, where they go. Same thing, cortical bulbar tract. The last part of our table for our indirect extrapyramidal pathways, where they go, where they decussate, how they work, and what they talk to. Which brings us to our cerebellum and our basal nuclei. Now, the cerebellum performs 4 activities, as do the basal nuclei. The cerebellum monitors intentions for movement. It monitors actual movement. It helps comparing command signals with sensory information, and it sends out corrective feedback. Now, the basal nuclei deals with 4 activities as well. This is initiation of movement, like I am going to start walking. Suppression of unwanted movements. It regulates muscle tone, and it regulates non-motor processes. This is attention, memory, and planning. So we can process the information moving to and from the cerebellum. 1, monitoring intentions for movement. 2, monitoring actual movements. 3, comparing those command signals with sensory information. And 4, connecting sending out the corrective feedback. So we see that the pons is integral here. Again, there's our relay center, the thalamus. So we have the sensory signals from proprioceptors and muscles, joints, vestibular apparatus, and the eye coming in to the cerebellum. We have our motor responses going back and forth, working to corrective feedback, and then the corrective contractions to fix whatever was being detected, and then following that response out. The last few slides deal with integrative functions of the cerebrum. This includes the reticular activating system that regulates wakefulness and sleep. It also puts our body into rapid eye movement, or REM sleep. So the reticular activating system is what deals with hormonal regulation, like the pineal gland for melatonin, that looks for stimuli like the rising of the sun or setting of the sun. It tells our body when to wakes up our organs, put us into a wake-up protocol, and tells us when to sleep, activating parasympathetic situation. Next is a coma. This is a state of unconsciousness in which an individual has little to no response to stimuli. What can cause this? Swelling of the brain, trauma, injury, etc. As far as the integrative functions of the cerebrum, we're dealing with learning and memory. This includes immediate memory, short-term and long-term memory, which brings us to amnesia. We have anterograde amnesia and retrograde amnesia. And what are the difference between the two? Anterograde and retrograde means that you sustained an injury. After that injury, you have no memory and you cannot build new short-term memories. Retrograde, so this one affects short-term. Retrograde is those long-term memories you can no longer remember before an accident or injury to the brain. For language, we deal with Wernicke's and Broca's. Remember this from AP1. Wernicke's is the association area, so this is the interpretation of language, interpretation here of spoken language, and Broca's area is our ability to speak, right? So this is a motor area that allows us to talk. So this concludes chapter 16. Please look to our third lecture.